we will talk about what are the different indicators and what is the understanding of the validation of the autoclave. We are not completely blindly trusting on the autoclave. We have to have certain parameters and indicators to cross-check the process of sterilization and efficacy of the autoclave. There are three means and indicator. One is the mechanical indicator, second is the chemical indicator, and third is biological indicator, which is the gold standard. In the mechanical indicator, you will check the temperature, which is written on the autoclave, that you will note the change in the temperature. You will see the pressure from your pressure gauge, and you will note the time. So you will see the parameters that are that you record over the autoclave as a mechanical indicator. Secondly, in the chemical indicator, we have 6 different classes or types of the chemical indicator. In the type 1, 2, 3, they are not very sensitive. They just tell us about that the sterilization process has been carried out and there is a change in the pressure and temperature, and then steam has been applied to the pouches. But the indicator or the class 4, 5 and 6 are more accurate, and they tell us about the TTP. T stand for temperature, T stand for time, and P stand for pressure. They tell us a story that the proper pressure has been applied for a given period of time and a proper time has been applied. These 4, 5 and 6 classes are mostly the internal indicator. Internal indicators are those which are placed inside the pouches, and external indicators are placed over the pouches to see to validate the process of sterilization. Now come to the gold standard of validation. The gold standard is a biological means of biological indicators. In the biological indicator, the most common and famous is you place a vial in the autoclave, and in the vial you have the spores of the Geobacillus stereothermophilus. So these spores are placed in the vial, and you autoclave this vial along with the other instruments. You remove the vial, and the logic and the science is very simple. You see whether or not these spores do grow in the next 24 to 48 hours or not. What you do, you break the vial. I will discuss about the structure of the vial in a minute. You break the vial, that is called the activation of the vial, and you place it in the incubator. In the incubator, the temperature is from 55 to 60 centigrade, and you place it for up to 48 hours. You may send it to the lab to culture, or you can just simply see the color. That is quite enough to tell you about the process of biological indicator. What is the science behind it? Please do consider the structure of the vial. You have a plastic vial, so this is plastic, which is bendable as well, and you have a glass ampule over here. This is glass ampule which will break. So this glass ampule will actually break when you when you apply the pressure on the vial, and on the upper surface you have the cap, and in this cap you have minor holes in which the steam penetrate. Here comes the spores which are placed on some paper. Here you have spores. What happened? You place this vial in the autoclave, and during the autoclave or sterilization, during the autoclaving and sterilization process, all of these spores should die. When all of the spores are killed, and you break this glass ampule, and in the glass ampule there is growth medium. The growth medium, after the breakage of this glass vial, will come to these spores, and when you place this vial into the incubator for 24 to 48 hours over the period of with the temperature of 55 to 60 degrees centigrade, and as you have destroyed all the spores, then after 48 hours the color of this medium will remain the same, and it will remain the blue because there will be no growth of the spores. But if you have using a faulty autoclave and it's not completely destroying these spores, then what would happen? When you will break this glass ampule over this plastic, as I said, this plastic tubing is bendable. So when you apply pressure to break it, you will directly, this bend, and pressure is applied on the glass and this break. After this break, the growth medium reached the spores, and if these spores are alive because of any malfunctioning of the autoclave, this spores will start growing and this medium will turn from the blue to yellow. If there is change in the color, this describes that there is growth in the growth medium by these spores, and these spores are Geobacillus stereothermophilus. You may send this vial to the lab. Obviously, in the microscope, you could be able to see the growth of the spores, or alternatively, if you clearly seeing that the color is changed, and this color is changed because of the acidic medium and because of the growth in the vial.